Hi,
Hi, I'm Christian.
I'm Emma.
and I'm Chinier.
I'm Ranjan.
Okay, so today we're going to talk about the fruit fly model of epilepsy and the effects of cannabinoid on that model. This is just like a little list of what we're going to talk about today. We're going to first talk about the project description, and under that we're going to talk about the significance of our study in public health, and then we're going to go through our background and talk about previous studies and the crosses we use. For methods, we're going to talk about specific aims and how we carried out each specific aim, and then we're going to talk about our expected results.
Thank you, Shinru. So, our project description. In this proposal, we are going to be using GAL4 UAS cross of Drosophila melanogaster to study how the inducement of seizures is affected by cholinergic neurons and the inhibition of them. In this study, we're going to use multiple methods of assessment, one of them being behavioral analysis. We're going to be looking at their body movement and see whether or not there is cessation of limb movement. And then we're also going to monitor the post-ictal state 2, which is essentially what happens after a seizure when the central nervous system is trying to go back to its normal state, which generally lasts around 2 to 3 minutes long. This particular field of study is very relevant to modern-day medicine, as fruit flies are known historically to be a great model for how epilepsy happens in humans. And we're also going to be studying how the effects of cannabinoid is a possible treatment for the reduction of the frequency of seizures and whether or not that has any human application.
So for some background, in neuroscience, clinical studies have focused on animal models like fruit flies because of the analogous neural processes. Epilepsy is a neurological disorder characterized by episodes of disturbances in neuronal activity called seizures. In prior studies, they figured out that an imbalance of excitatory glutamatergic signals overpowering inhibitory glutamatergic signals is a cause of a seizure. In recent studies, cholinergic circuits might be associated with epilepsy as a result of their role in modulation of neuron signaling, specifically excitability, synaptic transmission, and synaptic plasticity. In previous studies, they've used the cannabinoid CP55940 for treatment of Parkinson's disease.
So recent studies haven't yet showed that there are any CB1 receptors, specifically cannabinoid receptors in cholinergic neurons, but like we've seen, cannabinoids can attach to other types of receptors, and it doesn't have to be a cannabinoid receptor, so that's what the basis of this is that's the basis of our experiment.
So these are our crosses. We did 2 crosses. So for the first was for our first specific aim, so that we could image those neurons that are involved in the seizure, or the cholinergic neurons. So we took one fly line with our cholinergic neurons and crossed it with another fly line to be able to label those neurons with GFP, so that when we dissected the brain and visualized it under luminescence, we'd be able to see those neurons nice and lit up for us. So we took that fly line, crossed them, and for offspring was what the ones we used. For our second cross, we took that same line for the cholinergic neurons, and we crossed them with another line that would allow us to oxygenically stimulate these seizures in these flies. So those 2 lines, cross them, offspring, those are the ones that we induce seizures in.
We have 3 specific aims. For the first one, like Emma mentioned before, we used the GAL4 lines in cholinergic neurons and the UAS GFP line, and we crossed that and we got the offsprings. So basically, we
Looks like I'll edit it.
So basically, we after we crossed the flies, we made sure that we got to the flies and took out the first offsprings, the first generation, I guess you'd call it, or the F1 generation, because those for sure would have, one of those offsprings for sure would carry the line that we need. So, because, after the first generation, then it's like randomized, and not all the flies would express GFP in their cholinergic neurons. So after that, we dissected their brains, and then we put the fly brain, after staining the brain with that, we put the fly brain under a fluorescent microscope, and using imaging tools on a computer, we were able to get an image of the fly brain and the area of the brain that we're looking for, which is cholinergic neurons, as shown here.
So for our second specific aim, we wanted to visualize the behavior of the post-cell state, which is that state right after the seizure. So we took our flies, both male and female, and individually put them in these little petri dish containers where we were able to, once they were, moving around normally, shine yellow light on them to induce these seizures, start recording our flies, and then look for those things that would indicate a seizure, like Fisher mentioned earlier, the cessation of body movement, maybe a little twitching, maybe they're not walking properly. And then we continued to record those flies until they were up and moving around like normal flies. We did this a bunch of times so that we would be able to have a nice big data set of how the light is affecting these flies and what the seizures are looking like. We did this with both constant yellow light for 5 seconds and then a pulsing yellow light for 5 seconds, just to see if there was a difference in the way that we were stimulating these seizures with light, if that played a role in the way the seizures would be.
So for our specific aim 3, we treated the seizure flies with the cannabinoid CP55940, and we did this by moving the flies to an empty vial and administered it on a piece of paper in the vial because they need to inhale the cannabinoid. So former research has found that cannabinoids that serve as agonists for cannabinoid G protein coupled receptors in the human brain can be used to treat certain neural modulatory diseases.
Like Parkinson's. But we decided for our experiment, hopefully that prior study would also be able to translate to cholinergic neurons rather than dopaminergic neurons. And there's not enough studies to show that translates easily or nicely. But our studies did show that we need more knowledge on it, or just more experimental data on it.
So we've been lucky enough to trial these 3 specific aims under the guidance of Dr. Tulai, Dr. Gabroghi, and L.A. Daniela. In our first aim, we 100% we successfully imaged and labeled the cholinergic neurons in our fly brain by first dissecting the fly brain, which was then preserved in PBS, PBST, and formaldehyde, and then we used DAPI to mount the slide to neuroimage it. In our second aim, we performed a comprehensive behavioral analysis. Generally, we have our 2 types of seizures. We have our generalized tonic-clonic seizures and our absence seizures. The generalized tonic-clonic seizures are generally what people see in media with the movement of the limbs, and the absence seizures is usually what fruit flies present with. So this could either be no movement at all, staring, right, which usually represent focal regions of abnormal neuronal firing. And we saw this when as Emma mentioned, optogenetically, when we would present them with yellow light. In our third specific aim, we attempted to use cannabinoid to reduce the frequency of these seizures. We noticed that this wasn't effective in at least our methodology, but we are optimistic in the potential use of cannabinoid for the treatment of. Okay, but we are optimistic in using cannabinoid for human application in the treatment of seizure.
Especially because compared to its counterparts, cannabinoid can be an easier and more affordable type of treatment rather than the current treatment for Parkinson's and epilepsy, which is partly why we chose the experiment.
Especially with the ongoing beginning of the legality of different types of cannabinoids on the state and federal level. This could be an option in the future. For our further experimentation with this, we would like to try the cannabinoid treatment again, maybe this time with using it in the food instead of as an inhalant. That might not be a proper way to treat seizures in flies. But yeah, thank you.
Thank you so much. Have a nice day.
Hell yes! And Emma just suggested